I don't, for what it's worth.
Is it amino acid?
Interestingly,
Right. Many of these things have.
So it was very beneficial for.
my name is Matt Caberlin, and welcome to the OptiSpan podcast. Hey everyone, welcome to the OptiSpan podcast, coming to you live from the 2024 American Aging Association conference here in beautiful Madison, Wisconsin. I am thrilled to be joined by Cristal Hill, one of my favorite people in the field. I've known Chris, I think we first met at a Biology of Aging summer course in Woods Hole.
2014.
Yeah, so Cristal, you did your PhD work with Andy Bartkey,
Yes,
correct?
correct.
And then went to Pennington Biomedical Research Institute, did your postdoc with Chris Morrison,
Correct.
who I actually just had the opportunity to go out to Pennington a few weeks ago.
Good.
And had a great visit with Chris and the other people there. And then you've been at University of Southern California, Davis School of Gerontology for about a year.
Yes.
Building your own independent research position,
Yes.
assistant professor, tenure track there, and moving up the ranks. So first of all, congratulations
Thank
on all of
you.
your success, and just gave a great talk
Thank
at the
you.
conference. And so your research has I think been focused a lot on the role of protein and how protein interacts with hormonal signaling to impact health and longevity. And I know that's a topic of interest for many people who watch the podcast. So, so I can't wait to dive in and get your thoughts on that
Okay.
topic.
So you want me to start from like the beginning? Yeah.
So maybe you can start by just kind of summarizing how you got interested
Okay.
in this research and its relevance to health span and lifespan.
Okay. Initially, I was interested in high carbohydrates diets, mostly simple sugars, and high fat diets and how they impact the aging process. And so once I finished my work for grad school in the Barkey lab, I was presented with this opportunity to learn about protein restriction. And why would you want to do that? That was my initial thought. So my whole...
You mean like, why would somebody want to protein restrict,
Right,
or why
right.
would you want to study it?
Right. So no, not why would I want to study, but in most we hear that you should consume a lot of protein.
Yeah.
And that was completely new to me. And since I have been doing high fat diets and high sugar diets, why not go and understand how does protein quality or source or percentage, how does that impact these longevity or these conserved genes that we've been looking at for years? And so I took a risk of going in to do something I've never heard of. And I embarked on understanding how protein restriction improves health span. How does animals sense that they are protein restricted? And how do we leverage protein as we get older? And how does that impact hormones, as you just said, namely FGF21, fibroblast growth factor 21. This is a well-known hormone that is released from the liver under different conditions. Fasting was the first known way to increase FGF21, but through the protein restriction diets, you have an increase as well.
Is there any relationship between FGF21 and growth hormone signaling, or are they thought to be completely independent?
No, there is a relationship. So what has been shown from Steve Cleaver's lab is that FGF21 actually inhibits growth hormone. So those animals are actually a little bit smaller.
Which animals?
The FGF20. So there's a study of FGF21 transgenics that's overexpressed.
Okay.
Sorry,
Right.
And that's been shown to reduce growth hormone signaling or pathways within those animals.
Is that known how that works?
No.
It's interesting. Huh. Yeah, I mean, it got me thinking because, IGF-1 is responsive to protein, and IGF-1 production in the liver is controlled by growth hormone. FGF21 is also produced by the liver, but kind of opposite, right?
But there is data in that same paper that FGF21 overexpressors, these transgenic mice, they have lower levels of IGF-1 as well. And so they live long. And it is thought with that relation from FGF21 through the growth hormone pathways that they have this increase in lifespan.
I see. So let's take a step back because probably a lot of people watching maybe aren't familiar with that FGF21 study. So this was a study that overexpressed FGF FGF21 genetically,
Yes.
transgenically in
Yes.
mice, and it increased lifespan. It was a pretty big effect,
Yes,
right? It was like
huge.
30% or so.
In males and females.
And those mice had high FGF21 through development,
Mhm.
right? Do we know at this point whether that increased FGF21 during development is the developmental piece what's important, or the adult piece? Because, and I ask because, I mean, as I'm sure you know, right, the work from the Bartkey lab
Mhm.
looking at, I think it was one of the dwarf mice, right, that have low growth hormone signaling, or maybe it was the growth hormone receptor knockouts, that if you add the growth hormone back during development, you lose much of the lifespan benefit. So there's a developmental, there's a benefit into longevity from being deficient in growth hormone during development.
Mhm.
Maybe not all of it, but some of it. Do we know in FGF21?
I'm not sure. I know that they were transgenic embryonic stage.
Yeah.
So I'm not sure if there's a study where they have turned FGF21 on in middle age or not, and so, or constitutively turned on, and see if that adds the benefit. So I'm not sure, but it would be a great.
I'm guessing it hasn't been done, because there really hasn't. It's been sort of weird that study is several years old now.
Yeah.
And I haven't seen anybody really follow up on the FGF21 overexpressor,
right,
and its role in longevity. And it was a big effect,
yes,
right? So
a huge effect.
let's go back to the beginning. So I think the key observations here that you have made is that, so we've known for a long time protein restriction can increase lifespan in
Yes.
mice and rats.
Right.
And what you have seen, correct me if I'm wrong, is that protein restriction increases FGF21 expression in mice.
Correct.
And you need FGF21 in order to get the lifespan benefit from a low protein diet.
Correct.
In mice.
Correct.
And these are FGF21 knockouts.
Yes.
So can you talk through that experiment, the way you described it in your talk today, that protein restriction? Like, what does that mean when you say protein restriction, and how is that done?
So in a rodent diet, a mouse diet, you can kind of equate this to human 100%, right? And so for them, their intake is 20% protein of their macronutrient intake.
That's the standard
That's
mouse
the standard mouse diet. So what we do is reduce that protein content to a 4%, a 4 for that, yeah. And so that's really 5%.
And that's by calorie composition or weight? Or?
That's by calorie composition.
Okay.
And so we reduce that to a 4th, and it is not it's not restricting, or it's not removing protein. It's just reducing the amount. So right. I don't want people
I know it's a minor thing. So when you said you reduce it to a 4th, does that mean it goes to 5% of the caloric content?
Yeah,
okay.
Right. 5%. And so I always like to make sure that we say we don't remove protein from the diet. We just reduce the intake, or we reduce the macronutrient content, because you need protein.
Right.
Right. And so with this reduced protein content, we are allowing the body to maybe in a way have this increase in stress responses, because remember, FGF21 was a stress fasting hormone.
Right. And was that how it was first sort of described, as responsive to fasting?
Yes, it responds to fasting, and it responds to other macronutrients and other diet interventions now. But what it is that we are reducing protein content without malnutrition within the animals, but we also don't have any discrepancies or change the caloric intake. So there is no reducing calorie intake, and there's no malnutrition.
And mice on a protein-restricted diet will actually eat more, correct?
They do. Actually, they eat themselves. They eat more
become
food.
Hyperphagic.
They become hyperphagic compared to their wild type or their control litter mates on a normal protein diet.
Does FGF21 drive the hyperphagia? In other words, do the knockouts on the low-protein diet also become hyperphagic? They don't
respond. So the knockouts on either a low-protein diet or a control diet, they don't eat more or less than each other. So they have similar food intake.
So FGF21 presumably is in the brain somehow mediating this behavior.
Right.
There's a recognition that they aren't getting enough protein, or there's an imbalance somewhere that behaviorally then they eat more.
Correct.
Yet they don't gain weight, which is super interesting.
Correct. And so they don't eat more, they don't gain weight. These are the knockouts. And going back to the brain, you are correct that the signaling process for this whole feeding or food intake, feeding behavior, is a process that happens in the brain. And so for us and from the Morrison lab, understanding that this low protein diet induces FGF21. Also, this was shown back in 2009, I believe, that FGF21 crosses the blood-brain barrier, and that was also shown at Pennington. And so now we know that there's this liver-to-brain axis that controls food behavior in the context of protein.
Right. Okay. So what happens to the knockouts if you fast them? Are they sensitive to fasting?
I've never fasted FGF21 knockouts. I know they have been subjected to ketogenic diets, but as far as FGF21 knockouts, just a normal fasting diet, I've never done that. And I don't know of any data or any lab that has subjected them to a fasting or like an intermittent fasting paradigm.
Interesting. So if you fast, you will increase FGF21, right? If you give a little bit of protein, but they're still very calorically restricted, right? So in other words, is the increase in FGF21 in response to fasting because they are protein restricted, or is it maybe a different mechanism of induction?
So we think it's a different mechanism of induction. They're correlated, heavily correlated, or strongly correlated. And I will say that because we did a pair feed study. And if you're thinking about fasting and changes in food behavior, intake, as well as energy expenditure is what I'm going to get to. So when we did the pair feed study, we had one group that was control ad lib, eat whenever they wanted to. And then we had a low protein group that consumed ad lib. The third group, the pair-fed study, they consumed as much of low protein diet as the control group, and we still saw this increase in food intake. So there is and these are all wild type animals, and so there is a strong correlation with this increase in food intake, but it is not only reliant on the effects of FGF21. It's the low protein diet.
Got it. Interesting. Okay. So FGF21, again, it's really shocking to me that this hasn't been studied more, given that we know now it is both necessary and sufficient for longevity extension, at least in the context of low protein, right? That again, I'm just going to say it. I know you know this, but just so everybody gets it, right? So if you put mice and rats on a low protein diet, that increases lifespan. That's been known since the 1930s, right? That was the original McKay studies, was low protein.
Right.
That requires FGF21 to get that lifespan extension. And then more recently, there has been a study showing that if you just overexpress FGF21, a normal diet, that is enough to give you a big lifespan extension. Why hasn't this been studied more extensively in the field?
I think, we're trying to understand how FGF21 actually works. For one, a big problem with FGF21, it has a short half-life.
Yeah.
So going in the way of looking at it from the perspective of diet, your body's making the physiological levels of how much it needs. And so I think we're getting to that point or that place in the field where we can implement these different types of diets to understand the long-term effects of just physiological levels of FGF21. And because if you inject it, it only has a 30-minute to 2-hour half-life.
Yeah.
And that's expensive. So trying to understand, trying to do that, especially in preclinical studies, such as in the rodents, could get quite costly. And so I think we're just there now to understand how these natural interventions could give us this future data that we can understand how it actually works for a long term.
So, and so one thing that might be worth commenting on here is in the transgenic mice, I think you've told me that those mice have, I don't know, like 100-fold increase in FGF21, right? Is that something like, it's a very, very big increase.
High, yes.
And so how does that compare to the magnitude of increase you see in response to protein restriction or fasting?
So it's physiological. So it's constitutively expressed because you're eating a low protein diet, and it's just normal levels, and it's going to vary per animal, even with humans. There was the proof study where they actually looked at it as well.
Yeah, we haven't said that yet. But
sorry.
No, it's good. I'm glad you brought it up because it is important to say fasting or protein restriction will also increase FGF21 in people, right?
Right.
Yeah, but again, if I understood what you said correctly, you said it's physiological, meaning that it's it's induced, but it's lower than what's seen in the transgenic
of course,
mice.
much lower than what you see in transgenic mice.
So that's still a little bit of an open question then, is that level of induction sufficient to give you the big lifespan benefit that was seen in the transgenic mice?
Yeah, that's true. And even we haven't gone below the 5%, 4% of protein in the diet, but there are other labs that have gone down to 2, and it's not so great. So I don't know.
What do you mean by it's not so great?
The animals don't survive.
Oh, for lifespan.
Yeah, for lifespan, they become sick. And so is there a space in between 2 and 3? And I have not tested that. Maybe someone else has, but I'm not aware of those studies.
Yeah. So do we have any idea at this point for, let's just say that the reason protein restriction increases lifespan is at least in part because you're increasing FGF21, which the data are consistent with that. Do we have any idea what FGF21 is doing mechanistically to improve health span or lifespan, to the extent that you want to talk about it? I know you've got unpublished stuff. I'm not asking you to share unpublished data, but to the extent that there's published data out there to address this, like, what do we think is going on with FGF21?
So there are a couple of labs that's been trying to understand how, once it crosses this blood-brain barrier, what neuron population is it actually activating on, and how does that send the signal back?
And are we 100% sure that all of the FGF21 activity is in the brain?
For now, we are.
So that sounds like a no. We're not 100% sure, but that's what we think is going on.
Well, we're evolving. So, the data does point to it, even though they're, hypothalamus and hippocampus, and there's some papers that show that these tenocytes at the in the hindbrain actually have an effect. So
again, how does this work? So I should know more about Klotho than I do. I will admit it. But so Klotho, the beta-Klotho transmembrane receptor in the brain, is that right?
Receptor, they're FGF21 receptors, yes.
So FGF21 goes from the liver to the brain. Mm-hmm. That's where it binds beta-Klotho to create this
It binds
receptor?
Within this homodimer receptor. So there has to be a FGFR1 and a beta-Klotho receptor together to create a homodimer. And then FGF21. It's almost like a insulin and IGF-1 receptors. It's a homodimer. Mm-hmm. And so there has to be that co-receptor in order to elicit the cell response.
And is beta-Klotho expressed in the brain?
Yes, we actually deleted Klotho, beta-Klotho from the brain. And so that's how we were able to test is FGF21 signaling in the brain required to improve metabolic health. So before the longevity paper, we had another paper to show which tissues, in our eyes are trying to understand endocrine tissues, mostly that these tissues respond to low protein diet. Adipose tissue, you have a reduction in body weight gain, which is really in the adipose. And then how does that affect the brain as far as feeding behavior? And so we actually deleted beta-Klotho from those two tissues.
Yeah, so let's talk about this, because this gets to the question I was asking of, is it FGF21 activity in the brain that's driving all of the FGF21 phenotypes, right? Sure. So again, just to make it clear to everyone, what you did was let me take a step back. So the way this works, and you tell me if I get this right.
Okay.
So beta-Klotho is expressed in the brain.
Yes.
FGF21 is expressed in the liver.
Yes.
It crosses the blood-brain barrier, gets to the brain, but presumably can go to other tissues as well.
Of course.
But in the brain, it interacts with beta-Klotho to create this dimerized receptor, and that's the molecule that's actually doing the action, we think, right?
Mm-hmm.
Okay,
yes.
It's transmitting this signal. Okay,
right.
So you knocked out beta-Klotho in the brain.
Right.
FGF is still being made in the liver. It just, when it goes to the brain, it can't do its job because there's no beta-Klotho for it to bind to and interact with.
In the liver?
No, in the brain.
Oh, yeah, in the brain. There's no, so the two were a wild type and then a knockout model.
Right.
And so there's no beta-Klotho for FGF that's circulating FGF to interact with.
And okay, so one question is beta-Klotho expressed anywhere outside the brain that we know of?
Other tissues.
It is.
It is.
And in your mouse model, it was only knocked out in the brain.
It was only knocked out in the brain. And they had every other tissue was reserved or preserved for beta-Klotho expression.
Okay. Got it. Okay. So now tell us what happened in that mouse.
So those animals, they do not respond to protein restriction at all. So they do not have this reduction in body weight gain. They do not have an increase in energy expenditure. Their glucose metabolism is completely impaired, as well as their energy metabolism. So
this is all on a low protein diet.
This is all on the low protein diet, and the beta-Klotho brain knockouts. They do not look like the wild type animals on a low protein diet, which are the beneficial animals.
And are there any defects in a normal diet that you know of in those mice?
In the wild type?
No, in the brain knockouts.
So it's blunted on they look similar on every endpoint. They're blunted because remember, this particular study are in young animals. And so we stopped this study within 3 to 4 months of the study. So they're still presumably young.
Yeah.
But as you get older, we would expect to see the more detriment impact.
Yeah, and so we didn't talk yet about this, but the FGF21 knockouts on a normal diet, what happens to them? Do they have defects, or are they pretty normal?
They're pretty normal.
Huh.
And remember, these are young animals, so they're pretty.
Yeah.
Young.
Did you do a lifespan on the FGF21 knockouts on the normal diet?
Yes.
And
they actually live longer than the knockouts on the low protein diet.
So they are more or less like wild type mice on a normal diet.
For the longevity study. So we did see differences only in the longevity lifespan curve, but still their metabolic profiles resemble the same. So we have new data that we're going to be sharing very soon. What happened to those particular knockouts on a low protein diet?
Yeah, cool. Yeah, I know you've got some really exciting unpublished data that is starting to get towards mechanism. So we'll save that for next time around. But okay, so back to the brain knockout. So for the behavioral effects of protein restriction and the metabolic benefits of protein restriction in mice, those brain knockouts of beta-Klotho, which are presumably also brain knockouts of FGF21 activity, showed an absence of those positive responses.
Even that and plus they don't select for protein choice. Even if we try to give FGF21 back to the brain, they don't eat more or consume or select for protein.
Right. So again, what happens in wild type mice on a low protein diet, if you give them the choice, they will try to eat more protein. Right.
They'll try to consume
That's what
more
you
protein.
mean by select for more protein.
Yeah, they'll eat more protein.
And the FGF21 brain deficient animals are not able to do that.
They don't.
Now, what do you mean by give FGF21 back?
My PI, Chris Morrison, was very interested in behavioral studies and how animals choose or select, or what makes an organism choose or select a certain macronutrient or food product. And so we were able to do ICV surgeries. This is where you put a cannula, surgically insert a cannula into the mouse's brain, and we are able to administer pharmacokinetics or pharmacological doses of whatever drug that you're interested in. And so within the brain knockouts, we were able to do these surgeries, and we gave them back FGF21 signaling in the brain instead of.
And is that by giving them beta-Klotho? Because this was a beta-Klotho brain knockout, right?
We were able to do these brain surgeries within the brain knockouts in the wild type animals. So I only talk about the knockouts here.
Okay.
All right. So we were able to do ICV surgeries, and you're able to put a cannula on the brain of the animal, and you're able to give or administer drugs back to a specific location in the brain. And so we gave FGF21 FGF knockouts FGF, and they did not respond on selecting protein.
So these are the FGF21 knockouts, not the...
These are beta-Klotho, so I get that mixed up.
So, no, but I think it's important, right? Because if it's beta-Klotho, why would you expect to give FGF21 to the brain? Why would you expect that to do anything? Because it doesn't have to bind beta-Klotho in the brain?
That's what we're showing, right?
Okay.
Because we have that compared to the wild type group.
I see. So this is kind of a control.
Yeah, they're the control for the wild types, that everybody is on a normal diet. There's no intervention of a low protein or a normal protein diet. They're all on chow. And so what we do is make them feel protein restricted by giving them the FGF back to the brain.
Now I get
and
it.
they select bottle choices. So this was a liquid feeding choice.
So let's just make it clear. And you tell me if I get this right. So if you take wild type mice, you give them FGF21 to the brain, just injected into the brain.
Yes.
Those mice will choose protein over other macronutrients.
Yes.
Correct. And if they're beta-Klotho deficient, they will not.
They will not.
Okay, got it.
Right.
So FGF, so that's interesting. So increased FGF21 creates a choice for protein. So what happened? Was this ever done in the FGF21 transgenic animals? I mean, are they just like, they just really want protein? You know?
We don't know. We don't know that.
Because that would be the prediction based on this, unless there's some sort of attenuation or compensation or something, right?
Right. You would think that they would.
They're just like starving for protein. I like protein a lot. Does that mean I have high FGF21 in my brain? Because I want protein all the time.
You'll live long for the FGF21 is completely up. But that's a great question. They do have, if we think about the signaling processes here, right? High FGF21, low growth hormone, low IGF-1. They also have low levels of mTOR signaling. So that could be very. I never tried.
Yeah, it's an interesting question. Right. Yeah, to think about is it the behavioral component that's driving the lifespan effect, or is it the protein restriction? Right. So I mean, what are your thoughts on that? Because I know you're thinking a lot about, and I know Chris thinks about this too, the effects in the brain, and that's driving physiological, hormonal, behavioral changes.
Right.
So do you think it's the protein restriction itself, or do you think it's the brain-mediated consequences of protein restriction driven at least in part by FGF21 and beta-Klotho?
So it's all a signaling process, right? So you choose to eat whatever product. You choose to reduce this protein intake or your protein intake, and the liver responds to that. It's the brain actually telling the body what to do after it does not meet this macronutrient value, right? And so it's telling the body to, hey, consume more. And if you continue to eat less than what the body normally or naturally used to get, then you're going to have these metabolic changes and these fluctuations. Yeah. And so I didn't really understand it until after I left that lab, or not really left, but transitioned to my own lab, that there's this whole black box or this misunderstanding of, is it the chicken or the egg or the chicken? But really, we should just be looking at the whole circulatory or circling effect of food intake to brain and brain back to periphery.
Yeah. But I mean, this is really important, right? Because I think about this in the context of like the standard American diet, right? SAD, SAD, right? Because it's a first of all, it's something we've never evolved for, right? But we are we have too much nutrition, right? We have too many calories, but it's also for many people low quality calories, and the consequence. And so I wonder if, and maybe there's evidence, is it the case that people who are eating too many calories but are mismatched in their macros might actually have increased FGF21, but the consequence there could actually be to drive them to eat more, right?
And that is part of the protein leverage hypothesis, right? So there's this change in macronutrient selection or intake, a reduction in protein intake would drive these. But over time, the body adapts, right? And so you have this increase in FGF21. I'm just speaking in the low protein context here. You have this increase in FGF21, and that is supposed to bring back homeostasis into the diet. Now,
and by homeostasis, you just mean balanced macros. Yeah, that's at least as we understand it now.
Yeah, that's how we understand it. But as far as within our standard American diet, we think more so or quality versus what you're actually consuming. So if you're consuming proteins that are of good quality, meaning not so much saturated fat inside that protein, because most of it's going to be meat, then okay. But if you're consuming like the bottom of the bucket, just ground and full of fat meats, then it's not the best quality. You think of where it comes from, how it's cut to give you the best or the above average food product.
Yeah, and so how, do we have any idea like how protein quality affects FGF21? I know you were telling me something yesterday about what was it? It was about soy. This was not in the context of FGF21.
No,
this is
not.
About soy versus peas.
Yeah, pea protein. So
there are differences in protein quality.
Right, there are differences in protein quality.
Does that tie into FGF21 or is that?
We don't know yet.
Okay.
We don't know as far as like plant qualities, right? This is particularly because there was a paper from James Mitchell lab by Mike MacArthur. And what he did was complete or make these rations that were equivalent to meat protein sources and plant protein sources. And what he saw is if you just reduce the total amount of protein, you get these beneficial effects.
And this is in mice.
This is in mice. It's related to, they're correlated with FGF21 levels. Another interest is, does it matter what plant protein that you consume? There are some studies that show that soy is okay, but to some people, because like we all have different variants of certain genes, that we may not be able to consume soy like other populations can. And so does that create an inflammatory response? And if so, should we change or should we alter our plant sources of protein? Because that is one thing that vegans or vegetarians, they have to consume more of these products or plant products so they can match their protein intake or balance. And so if they are one of those individuals that cannot or they do not know if they have these issues from like soy, then they continue to have it and then they have these immune responses that are associated with the intake. And so we have to now think about those individuals when we're making these like recommendations.
So some individuals seem to have be predisposed to an inflammatory response to soy protein.
Right. Everybody cannot have it. And so we have to understand where we can make way for those that cannot, what other sources are available for them to consume.
Got it.
Yeah.
So, okay. So that was a little bit of a tangent, but so I think, what do we know about FGF21 in people? So it's induced by fasting, seems to be induced by protein restriction. That seems to be shared.
Yep.
Do we know, does it drive this food preference?
We don't know. I have not come across any data that shows that FGF21 drives people to eat or to...
You'd expect it to drive them to want protein, right?
To want more protein.
What about variants? Are there variants in FGF21 in people?
There are variants in FGF21. There are variants in FGF21 in people. There are gene variants.
And do we know anything about those variants and behavioral or phenotypic consequences?
They're mostly associated with cholesterol levels.
Oh, interesting.
Yeah, so it's more the lipid metabolism pathways.
Or could be driving the preference for the kind of food they're eating that's affecting cholesterol levels.
It definitely could. And so that was part of the proof study that I was mentioning earlier. They looked at those endpoints within those individuals away from just the FGF21 levels. What was driving or what was associated with that variance in FGF21.
So there are some people, so there's obviously going to be variation in individual expression of FGF21, but there are certain genetic variants that decrease FGF21, increase it. Do we know how those variants are affecting the expression level and activity?
I think they're more so in the sequence levels, sequence of the variants.
SNPs.
Yeah, SNPs in the gene. And so by that, how effective is it for an individual? How much can a person respond based off of a diet or intervention? Will they respond like the B6 mouse, right? You give them low protein diet, everybody goes up, right?
Right.
And so how much they respond to certain interventions is the key here. So understanding how those SNPs may be in individuals will give you better understanding of how to give them a diet or a new diet paradigm, which is really interesting because now, they have personalized medicine, but now they're going to personalized nutrition.
Yeah.
Or precision nutrition.
That's the goal.
Yeah.
We've still got a long ways to go.
Yeah. I think, NIH is doing a great job with the All of Us study because that's going to give us some insight. How do we help people on a independent in an individualized level.
Yeah.
Because while calorie restriction may work for some, it don't work for all.
Is FGF21 something you can measure easily in people?
It's a ELISA serum test.
But
Serum
is it?
plasma.
Do you know if it's available like
Yes.
as a laboratory test or is it a research test right now?
I think I'm not sure if it's a laboratory test.
Like, I'll look and see if LabCorp. We'll add it to the show notes. I'm just
Right.
interested to know if it's something that
Yeah, I
can
know
be routinely measured now.
You can measure it. I know the tests are there because I see them, the human kits when I'm ordering for mouse or
Okay.
rodent. Yeah, so
Okay.
they're there. I'm just not sure if they test it. I'm
Yeah.
pretty sure they test it in the hospital though. You can ask for it.
Okay, so
I'm
it is
pretty
probably a
sure,
laboratory.
Yeah, I'm pretty sure as much work has
clinical
been done.
test.
Clinical test,
Yeah.
right.
Huh.
I'm pretty sure that you can order or ask have it your levels
That would be
tested.
interesting to know.
Right.
Yeah.
Right.
Is FGF21 affected by exercise? Do we know?
There's minimal work that shows FGF21 is increased with exercise. So it's still out for debate how much and what type of exercise increases FGF21.
Huh.
But I would think it would go up, since exercise does induce some stress responses, but how much effective of the FGF21 increase is going to model or remodel some glucose or some type of metabolic endpoint is questioned.
Interesting. Yeah. So it seems to me like there's a lot more questions than answers right now, I got to say,
Yeah.
which is a good place to be if you're running a research lab. I still think, to me, it's very intriguing, right? This connection between FGF21 and beta-Klotho and alpha-Klotho. It just seems like there's got to be, like, the idea that alpha-Klotho would be a longevity factor and a, brain, affecting brain function.
Mm-hmm.
Independently of FGF21 completely seems that seems like very unlikely to me.
It seems weird that we didn't look for it, huh?
Yeah, I mean, I think there's just experiments to be done that are important experiments, and I think that there's some pretty clear things to do that should be high priorities.
Right.
So. You should write some grants.
I know you gave me a lot of ideas. As a first year assistant professor, you're like, okay, what's next? And I think I got them today.
Yeah. So actually, one other question I wanted to ask you is, how big is FGF21? Is it like a small peptide?
It's small
or is
peptide, 19 kilodaltons.
And is it something that can be taken orally? And I'm not suggesting anybody do it, but I mean, there is there's a lot of interest in these bioactive peptides now.
I'm pretty sure it can, but the wild type form of it is only 30 minutes to 2.5 hours.
For the half-life. So it's very short-lived.
It's very short-lived.
And is that because there are certain proteases that target it?
Yeah, I think there are proteases that target FGF21 and break it apart, break it down, right? And so that's the problem. So now they're coming out with
stabilized forms.
They have these isoforms that are more stable, and they're shown to be pretty effective lately. They're putting them into clinical trials now, or
for what?
For human.
For what conditions?
Oh, metabolic conditions.
For diabetes or obesity?
Diabetes, obesity, and it was actually shown years ago that it only worked in obese patients.
Worked meaning what?
Meaning to reverse the metabolic detriments of a high-fat diet, obesity.
Does it, so in mice on a high-fat diet, does it make them lean?
It makes them much better in the sense of it reverses the impairments of diet-induced obesity.
Does it make them lean, though, the metabolic effects?
No, they lose weight. They actually lose weight.
Okay. And are they wanting
remember, it's part of this whole energy metabolism, lipid metabolism push here. So they reduce their adiposity. They lose body fat.
So, we haven't talked about the metabolic stuff, but so FGF21 rewires lipid metabolism, among other things. What do we know about that? How does that work?
So from our studies, we see that it actually increases de novo lipogenesis. And so what that is this change in lipid metabolism to be more active, and your body actually uses those free fatty acids. Okay. And so going towards that route, this de novo lipogenesis, this fat breaks down, and it actually, changes the whole profile of that fat.
And I think some of the amino acid restriction stuff can also have that metabolic effect, right? So I don't know if anybody's tested it, but a plausible mechanism would be the reason why amino acid restriction has that effect, because you're turning up FGF21, and that's all downstream of FGF21.
Right, exactly.
Okay, so FGF21 is being tested or has been tested for obesity in people. Is it used clinically? Is it approved?
No, because the half-life is still
okay. So it's just not a feasible.
Yeah, they're working on that now. So this was a couple of years ago when this study came out that we're trying to understand how to have a long-acting form.
So this would be interesting again in a lot of ways, right? But it would be different. So does FGF21 administration in the context of diet-induced obesity in mice, does it change food consumption, or is it just changing metabolism? Because in protein restriction, they actually eat more calories, but they don't gain weight.
Right. I don't remember the food intake data.
Okay.
This was a while.
That would be interesting, right?
I don't. It's a diabetes, a journal diabetes paper. I remember it when it came out because I was in grad school and we were discussing it then. And it's crazy I got into it now.
but I mean, you might think that FGF21 therapy could actually increase appetite.
It would be nice to see if those in that mouse study, if they even select for different macronutrients after you give them FGF21.
They probably didn't, but
they didn't.
But your prediction would be they would prefer protein over other types of food. So if this actually works in people for obesity, what you might get is an obesity drug that encourages people to eat protein.
Well,
which would be very different than the GLP-1 agonist,
right? But that's the pharmacological way, right?
Yeah.
And so, why just not eat less protein and have normal levels of FGF21?
I don't know. Yeah, I mean, again, you could restrict protein. I think the concern with restricting protein in people, right, is whether or not it's still an open question. Does it have negative consequences on body composition, bone density, lean mass? I think it's TBD, but there's reason to be concerned about that. Although, Gary's talk
that
on
was
mice,
so interesting.
So for those of you who aren't at the meeting, Gary Churchill from JAX gave an amazing talk on caloric restriction and intermittent fasting in mice, which maybe I'll dive into in a future episode. But the intermittent fasted mice did not show a negative change in body composition, which I thought was interesting. So, I mean, and I don't think the protein restricted mice necessarily show a negative, although nobody looks at bone density. They just look at weight normalized lean mass.
Right.
So
and that
take it for what it's worth.
Yeah, I think if we have DEXA, it'll be very
that would
powerful.
Be important.
Yeah, because we just do the NMR to look at the composition of body fat and lean mass, and we do the calculations, but to see the bones actually would be very
and I mean, for frailty, this is my speculation, bone density is more important in people than it is in mice. Like I think the way mice live in the laboratory, in the cages, if you do like CT scans after death, you'll see fractures, but I don't think it limits their life the way it can in people, and certainly quality of life. So
I did like the part, especially for some of as we're aging, some individuals have a problem with gaining that weight, especially at older age, that is a little bit more healthier. And so, I mean, just from a personal, I tell my parents all the time, don't worry, because I'd rather you have that little extra fat to protect you if you fall versus you be thin and frail.
Right. And the epidemiology certainly suggests that being a little bit overweight in your I don't know when it starts, 70s, maybe. There's a reduction in all-cause mortality. So I think it's complicated. Biology is complicated.
And I just put out here, protein restriction up to a certain age, not lifelong, like not starting in late life and going certain age if you start early enough. So I'll leave it there.
So you're a fan of protein restriction for people.
To a certain age, because we do not want to create any insults of malnutrition. A big question that we have not answered, and I get asked this question all the time: why do we give older or senior individuals more protein, right? And so I'm wondering, are you trying to supplement for some type of insult or impairment within the digestive system that has changed over the years, that they're not breaking down the proteins, they're not absorbing?
I don't think we know.
We don't. And so that's why I was like,
but I mean, I think the rationale for that, is really driven by the expectation that a higher protein diet is going to maintain lean mass and bone density better than a low protein diet. Yeah. And we know especially with older adults, in their last decade of life, they tend not to eat as much.
Right.
So it's just trying to make sure they're getting, enough protein to maintain muscle mass and bone density, as much as possible. Even though I'm a proponent of pretty high protein, at least for me personally, I think a lot of that is just driven by expectation. I don't know how good the data is that force feeding protein to people actually has a positive impact on body composition. Yeah. It just fits with the way we think bone density and muscle mass respond to nutrition. So,
right. I also think, I use this analogy a lot as well. If you think of like high football athletes, people that consume a lot of food, let's just say The Rock, right? The Rock eats a lot of food. But he has the muscle mass that's going to actually utilize it and burn it.
And presumably people with more muscle mass are, at least in general, less likely to have negative metabolic consequences to what they're eating, right? Because the muscle is a really great sink for soaking up glucose and things like that.
Let's say he stops doing all of that and he continues to eat the...
Yeah, well, I mean, that's pretty common, right? Yeah, and then if people don't turn their appetite down, so, that
There's
doesn't...
that whole, understanding of what really switch turns or what happens.
Yeah, I mean, and again, this is total speculation, but, thinking about FGF21 as a therapeutic and some of the challenges people have with the GLP-1 agonists when they come off of them and their appetite, skyrockets right back up. I wonder if there may be a place with the FGF1 therapeutics to help with that if people don't want to stay on these other weight loss drugs. Again, who knows, right? We'll see where that goes. But it's interesting to think about the therapeutic applications of modulating FGF21 levels. So anyways, going forward, I think there's a lot of really interesting biology here, and we can talk about specific experiments that that I would like to see done. And if I was the the geroscience funding czar, I'd throw a bunch of money at this because I think there's a lot of really interesting stuff here.
I think we need to, like said today, we need to really make sure that we are detailed in how we do these or carry out these studies.
That was one of the things Gary said that's really important. And there's a lot of people who do sloppy caloric restriction or fasting experiments in mice. And Gary was very clear. It's really important how you do it, when the animals are fed, how much they're fed. Make sure you measure all of this stuff.
I agree with that. I think that's important. So,
Cool. Well, thanks, Chris. This has been a lot of fun. Yeah. I always love talking about protein and FGF21. Maybe next time we'll talk about the work you did with chickens and cows and what was it, priming the turkeys? So we'll go there.
Look, I've worked on all farm animals, hence now I love mice.
Awesome. All right. Thanks everyone for watching this episode of the OptiSpan podcast. As always, any questions or comments, please leave them below. If you're not yet a subscriber, make sure to hit subscribe, and I hope to see you next time on the podcast.